Hey bioteam. So you guys have learned that proteins are long polymers made up of smaller monomers called amino acids. And all amino acids have a couple of similarities in their structure. They all have a central carbon that's bonded to an amino group and bonded to, on the other side, a carboxyl group. Where amino acids can differ then is that there are 20 different types of variable R groups that an amino acid can have called side chains. And these amino acids can connect together. So we'll have a second amino acid here through dehydration reaction. The hydroxide group from the carboxyl side of an amino acid can go through dehydration synthesis with one of the hydrogens on another amino acid. And so we'll show that happening here. And you'll notice that a water molecule breaks off. And what's left over is two amino acids connected by a covalent bond, which biologists call a peptide bond. And so we'll show another peptide bond forming here. Again, in dehydration synthesis, water molecules break off the longer polymer. And so through a series of peptide bonds, a series of dehydration synthesis, a longer strand of amino acids can get synthesized together into a protein. And so proteins then can also be called polypeptides, just many peptide bonds strung together. So there's three types, three general categories of amino acids. There are amino acids with non-polar R groups. There are amino acids with polar R groups. And then there are amino acids with ionized R groups. That is, they have an entirely positive or an entirely negative charge on them. And so the type of amino acid and the type of R group on the amino acid will affect its position in the longer protein chain. So for instance, non-polar amino acids, because they are not charged, they are hydrophobic. That is, they don't interact with water molecules, which are charged, which are polar. And so as a result, non-polar amino acids tend to bury themselves toward the inside of a strand of protein away from water surrounding it. And so the way you can identify an amino acid as nonpolar is if you look at the very tip of an amino acid's R group, or the very tip of its side chain, you'll see that it's made up of carbon and hydrogen bonds, which are, again, nonpolar bonds. Carbon and hydrogen have very similar electronegativities. The next type of amino acid is a polar amino acid with a polar R group. And so the way to identify a polar amino acid is at the very tip of the amino acid's R group, there will be an oxygen-hydrogen bond, which is polar, a nitrogen-hydrogen bond, or in the case of cysteine, this is just one of the 20 amino acids, there could be a sulfur-hydrogen bond. All right, and then the last type of amino acid is an ionic or a charged amino acid. And since an ionic amino acid is completely charged, either positive or negative, it is also hydrophilic. It attracts to polar water molecules, which have charges. And so these can be oriented toward water molecules, or they can form something called a salt bridge by bonding with an amino acid of the opposite charge. Okay, so for example, this amino acid here, this green one, has a positive charge to it at the very end of it. And this purple amino acid has a negative charge. These two amino acids could potentially bond to each other. And so the way to identify an ionic amino acid is probably the easiest. You just look at the tip of its R group, and you can identify a positive or a negative charge. And that's it. At this point, you guys have some practice problems. Best of luck.